I assume people are out there. Have a bright light. I wanted to welcome you to the Luminary series. My name is Gary Schindel. I'm the Administrative Vice President for the NSS. And Dave will introduce our Luminary speaker.
Good afternoon, ladies and gentlemen. Our speaker today is Dr. Diana Northup. Shortly after Diana began caving at the University of West Virginia, she wisely decided to combine her enthusiasm for cave exploring with her lifelong interest in science, and in particular, the life sciences. Through many years of research, publication, teaching, and lecturing, Diana has established herself as one of the world's foremost experts on certain types of life in caves, including microbes and extremophiles. Indeed, Diana's research into particularly hostile and savage environments will very likely inform future explorers as to appropriate places to search for life elsewhere in our solar system. Because of her professional expertise, impeccable reputation, wit, charm, and lucid explanations, Diana is a sought-after speaker not only for professional symposia, but also for general audiences. For example, she has appeared on Discovery, National Geographic, documentary television for the BBC, and on the Nova Network. Please join me in welcoming your 2015 NSS Luminary, Dr. Diana Northup.
So it's actually a lot of fun being an expert in slime, as you'll see in the next 50 minutes. So was very sad when I looked at several of the other Luminary talks to realize that I didn't sneak away from school and go into caves when I was in grade school or junior high. Mine started a little later, but you're going to see the journey that it took me on to be able to develop my life's passion for extremophiles. So caving and caves have given me a couple of really important things. They've given me family, including 2 husbands, and lots of really neat friends, and trillions of microbe friends, many of which you're going to get introduced to today. So many of you know Bill Jones and Gordon Smith, who are responsible for my being in caves. If you have never seen it, this is Bill Jones in the upper left and the upper right, and in the bottom right is Higginbotham's number 1, which is, we think, one of the first caves that I went into. And I had a very boring childhood. And so when I joined the West Virginia University Outings Club at 18, and Bill took me in this cave, and the first thing that happened was I fell in over my head in water, and I thought that was the coolest thing that ever happened in my entire life. So that was my introduction to caving and the beginning of West Virginia caves. So I was entranced by leaving the sunlight and going underground to see what I could find. This is me at 18 in Scott Hollow Cave with Bob Reitmeyer. And here are many of the West Virginia cavers, and I think Ray Cole will recognize himself in a couple of these pictures. That's him climbing the rope, and Dave Hubbs is in there, and Frank and Judy O'Hara, and Bill Jones up in the upper left again. Lots of the West Virginia cavers from back in the 1960s. But hundreds of feet from the entrance of Scott Hollow Cave, I saw this little guy, little pack rat, just wandered around in the darkness, and I was like, that is really amazing. How is he doing that? How can you live if you live in this environment? And I became entranced with the idea that life can exist in this very hostile environment. And so I determined that I wanted to study biospeleology. And one of the first things I did is I wrote a letter to Dave Culver, who was beginning his work in the West Virginia caves on invertebrates, and said, I want to be a biospeleologist. Well, unfortunately, physics intervened, and so I abandoned that plan for a few years and actually moved to the University of Illinois, where I got a library degree and became a research librarian. And you may recognize Katie Sukup in the upper right there in West Virginia. And this is me in Pless Cave, which I understand had a gasoline spill and is either closed off or being rehabilitated. This is Sue's bathtub. So Illinois caves were really cool. And then I moved on to the University of New Mexico in 1977 to be a medical librarian there. And at the NSS convention at John Shelton's Ranch, what year was that, guys? 88. That's where I met Kathy LeBoy, who was the cricket lady. And that's Kathy on the right, and she mentored me in a lot of how you catch crickets. What she didn't understand was that she has thousands of crickets. We have hundreds of crickets in Carlsbad. So that is Ron Curbo in the upper part, and he and I set pitfall traps back in left-hand tunnel, and we didn't think we'd catch anything. We came back the next day, and the trap was full of crickets, so they were just hiding from us. So I did several years of cricket studies based on some of the work that Dave Culver did, and looking at some of the ecology and the evolutionary predictions, and looked at crickets like this one that are not as cave adapted, and also looked at crickets like the longipes that are very cave adapted. And I don't know if you know, but if you're trying to speciate and determine what species of cricket you have to press on the male abdomen and get it to avert its penis. And then you fix it with ethanol. So one of the cave rangers came up to me in Carlsbad and said, "You can press on my tummy." And I whipped out my syringe and said, "I don't think you'd like the after treatment." He quickly beat a retreat. So many years of cricket research taught us a lot about the crickets. However, this is what it does to your field helper. I even had one person who could sleep in 60 feet of bat guano. So, and this is the 60 feet of bat guano in Carlsbad Cavern. And that's us serving in our trap sites. This is my first case of histo, because we didn't know that dust masks are not HEPA filters. So that was my first unfortunate thing. The other thing is, Cal Wellborn and I did a survey every 28 days of 9 sites in the guano throughout Carlsbad Cavern, and you had to count the mites under a microscope. I got this frantic call from Cal in July saying, "Cut the sample size." It turns out there are 10,000 mites per cup of guano in July in Carlsbad Cavern. And you may have noticed this work has never been published. So, about that time in the late 80s, I met the gentleman on the left, Cliff Dahm. I actually took a microbial ecology course from him. And he handed me a bunch of vacutainers, and he said, "Go get some water out of Lechuguilla Cave," where I was doing an invertebrate census for the Park Service. And I went and got the water, and we started looking at it, and we saw the critters you see here on the bottom right, a Celebaria and some kind of what we call a prosthecate bacteria. They look like sperm on testosterone to me. So this was like opening the door to Wonderland. And about the same time, Kim Cunningham, in the upper right for you, was working in Lechuguilla doing all kinds of amazing science studies in Lechuguilla, and he encouraged me to start looking at the microbiology of Lechuguilla Cave, which I did. About the same time, I met Penny Boston and Mike Spildy, and we established the slime team. So this is the start of a long tradition of slime. So the subsurface life in mineral environments, that's our t-shirt, designed by Bonnie Kernock, and us in Hawaii pursuing slime. Also at that same time, Will White and others organized the Breakthroughs in Geomicrobiology Conference. This was an incredibly seminal conference that really provoked the field of geomicrobiology of caves to really expand and just skyrocket off. And we became interested in pursuing this in Lechuguilla Cave. So we were interested, at that time, we were still in the stage of going, who's home? And just learning who was there. But we also wanted to know, can they dissolve and precipitate rock? Are they rock eaters? Because this was an important part of geomicrobiology. And then a question that I'm still pursuing is there an indigenous subsurface biota like there are invertebrates? So that was one of the things we really thought of. Also in the mid-90s, I started a PhD at the University of New Mexico. And there was just one problem with this, and that is Cliff Dahm, my PhD advisor, who I had sent in alone to die in Lechuguilla, and he came back very bloody, said, you need to do it on Lechuguilla Cave. I'm terrified of heights. So this was a problem. So there I am, Bob Beecher, documenting all the worst moments of my life. He's very good at that. And so in order to be able to deal with being afraid of heights, I got people to sing me up the rope. And as they faded out, and as I approached the top, and there you have it, the slime chorus. You now have a name, guys. So that's how I conquered that part. But there were a few other rocky starts to it. And I want you to appreciate the next 5 slides. This is how Penny introduced me 3 years ago at a talk for her departmental seminar series, which she was not at because she was lecturing for me. So she left this introduction with John Wilson of the Department of Geology, introducing me, her wonderful friend. So we used to do a lot of work on spiders. So Penny would go dropping them in the poison. Bah! I swear I can hear them screaming. I think it's wrecking my karma, not to mention the cricket incidents. Remember the penises? All right. Okay, so I have to get the AV. Hello, it's dark down here at night in big caves like Lechuguilla. They turn all the lights off to go to sleep. Sometimes they get cold and have to put on other clothing, like, for example, a balaclava to keep their heads warm. It's really hard to know what's going on. But things don't always turn out as planned. What happens in the cave stays in the cave, or maybe it doesn't. This balaclava doesn't seem to fit very well. How can I be my usual stylish self? And that is the little told story of my underwear on my head. Yeah. So my adventures in Lechuguilla Cave included many dear friends who put up with all of this. You can see wide-eyed Bob Beecher, who has rescued sleeping bags out of pits in Lechuguilla for me. Stan Allison, who eats all the food that I decide I can't eat. And Larry Mallory, who some of you may know, who did the studies on various cancer-curing agents out of Lechuguilla Cave, Jim and Val, and Debbie Beecher. So we had a lot of adventures there. And what we were trying to establish in Lechuguilla and Spider Cave was looking at these bugs that are actually in these ferromanganese deposits that you see in the upper right, the reds and the browns and the blacks. So there was a punk rock zone underneath, and so we developed a model that basically some of those sperm on testosterone, those prosthecate bacteria, are down in the punk rock, and they are able to mine the rock for reduced iron and manganese. And when they strip electrons off, they actually produce acidity, which is what helps to create some of the punk rock. So rather than just having a corrosive air model, we also had some of it happening with the assistance of microorganisms, and Penny designed a really wonderful set of experiments where we were able to inoculate cultures and show that they can actually precipitate manganese and iron in culture. We also, at this time, I started working with Leslie Malim, who I had met in grad school the first time with my master's, and she is a geologist, and it took me 10 years, but I finally got her into doing cave research. And so we started working on the pool fingers of the Guadalupe Mountains, and here she is in, I believe, the Rattlesnake Room? No, this is Hidden Cave. And we're looking at these pool fingers that hang down from shelfstone and looking at how the role of microorganisms has developed in developing these pool fingers. So here you can see the interior of a pool finger, which has been acid etched, a technique that Leslie came up with. So you can see that the interior is actually full of microbial filaments, including what we call microchoia. These were named by Penny. And the microchoia look like a choia, a dead choia in the Southwest desert. And our life goal is to figure out who these guys are. And we actually have found an environment where we get heaps and heaps of them. And this is, if you heard Leslie's talk, or will hear her talk this afternoon in the biology section, these are some living pool fingers in Germany that some German cavers have helped us access. And those are very slimy and wiggly, so they're absolutely perfect for our study. This new paper that you see on top will be coming out in a couple weeks, Scott Engel tells us. And it is full of these microchoia, living examples. So we're ripping the DNA out of them as we speak. So part of the work I did in the ferromanganese deposits was to take DNA out of this and sequence it so that we would know what was going on. And to do this, I went and did a sabbatical in Norm Pace's lab. How many of you know Norm Pace is a caver who discovered Oasis Pool? So he is a caver also. And after about a month of trying to get DNA out of the ferromanganese deposits, he said, enough of this crap. It's not working, and took me to Parker Cave in Sulphur River, which was an amazing experience. We took along one of his grad students who it turned out is deathly afraid of cave crickets. And I don't know if any of you have been in Parker Cave, but it's full of cave crickets. So we spent the entire time going, Sue, keep your head down, don't look around, keep going, to get her through this. But that introduced me to the joys of sulfur caves. So, and a couple years later, Louise Hose came up to us at an NSS convention and said, you have got to see these. You have got to see these snot tights. And so she invited us on an expedition to Mexico, where we were able to start looking at Cueva de Villaluz. And here you see myself and Penny and Kathy LeBoy and Louise Hose. And we did a bunch of expeditions down there. If you have not heard of it, it is a sulfur cave, and it is about in the state of Tabasco, near the Chiapas border. And what you see when you come out is all this white stream that's coming along. And this white stream is full of elemental sulfur, which is really cool. It was, the cavers found it by asking the locals about caves in the area, and they went, azufre, azufre. And by the way, many people think a biologist named snotites. It's actually Jim Pizarowitz, is my understanding, that named these. And the cave is full of life. Whenever I show this in my class, I have a lot of arachnophobes who are like, no. But the cave is full of this amazing bounty of different spiders that are in there, and they have egg cases everywhere. And then I want you to look at the upper left for you, and in the tarsi of that diving beetle, do you see the fish? There is a fish jump where the fish, just like salmon, jump up this rock wall, and if they don't make it, they are dinner for the diving beetle waiting at the bottom. That diving beetle over on the right is covered with sulfur bacteria. And so they are mobile microbe hotels, if you will, which is a really cool idea. And the crab that is down here in the lower left is also covered with sulfur bacteria. So everything that is living in this cave often has some association with the microorganisms. So if I thought Lechuguilla was a wonderland, this was way beyond that. So we started really doing a lot with here. The fish are somewhat troglodytic. There's a German group studying these. They have on their eyeshadow for you. If you notice, they've dressed up. And here's another one of the diving beetles. And then in the upper left is a green biofilm in total darkness. So we're looking at some of the microorganisms that are present in that. And then over the various gas wells, you see the elemental sulfur. So the cave is so rich because of these various hydrogen sulfide vents that come into the cave. And your gas monitor goes off at 10 parts per million, telling you should get the hell out of here. And Penny and I have been back in an area that has pegged the meter at, I think, 240 parts per million. So it's a really dangerous place to be. So do you know what the first sign is that you're dying of hydrogen sulfide? Your peripheral vision goes away. So we were crawling through one of the crawlways, and I'm crawling along, and suddenly the lights go out. And I'm like, that's it, we're dead. And then somebody came around the corner, and I realized my light had just gone out. So one of the things that happens is you get pretty stupid in here because your respirator doesn't protect you against the carbon monoxide, just the hydrogen sulfide. And this is what you look like when you get really stupid. This is why my husband Kenneth's role is to basically take Penny and I by the bootstrings and say, time to get the hell out of here, ladies. He keeps us safe. So we've done a lot of investigations in here, and because around these various inlets back in the salon are the snotite heaven and snotite purgatory. So these are some actually really almost brand new pictures of snotites, one of my favorite things. And so they drip sulfuric acid. And one of the interesting things was when we did the NOVA filming down here, the NOVA producer just couldn't be bothered with the fact that he was getting some of the gypsum pH 0 paste under his knee pad. Couldn't be bothered to wash it out. So the next day he had a nice bloody hole in his leg that didn't do him any good at all. The other thing that happened is during one of the trips, Doug Soroka, I think we left him alone a little too long, thank goodness. And he started digging around in the bottom of the stream, and he discovered phlegm balls. And by the way, this is the centerfold for the January 2000 National Geographic issue, the only time I'll ever get to be a centerfold. And this is what phlegm balls look like. Perfect for after lunch, right? You can just imagine your worst cold hacking that up. That is actually, think of it as a microbial house. So we build our houses out of stone or wood or concrete. They often build a house of slime. So all that snot you're looking at is basically protective for the microorganism. It does all kinds of things for them. It's the reason if you have a catheter in and it gets colonized by microbes, and they produce all that protective snot. It's the reason antibiotics will not cure you. So understanding some of this is an important thing. And this, ladies and gentlemen, is the interior of a phlegm ball, courtesy of Mike Spildy and myself doing SEM. So you can see them waving at you. So there are lots of cool organisms present in there. The other thing that research did for me is it introduced me to the idea of talking to the press. And this is truly the best way to embarrass yourself beyond belief. So the very first interview I did was with a magazine called Buzzworm, back when I was working on crickets. And I had 102 fever, and I should have known not to talk to the guy. But when the article came out, I was quoted as saying that there were marauding red-brown crickets taking over the lunchroom of Carlsbad Cavern. That wasn't the worst. So if you look at this quote, mysterious organisms eat cave walls, vexes scientists. The next sentence basically said that I had discovered non-carbon-based life. I cannot tell you how many calls I had from people in the biology department, my department, going, what the hell are you talking about? So I learned that one has to be careful what one says to the press. And so we went on to do bigger and better things like the Nova Mysterious Life of Caves, which came out a whole lot better. I don't think we have any hideous stories out of that one. Although they did leave me hanging on the rope at Lechuguilla on the long side for half an hour, which is enough in an agoraphobic to create havoc. It was not a good day. And also, if Peter Jones is not here, so he can't appreciate this, but my safety officer was Dave Jones. And Dave thought the way to deal with somebody who's afraid of heights is to scare them. So he would snap the rope on the edge of Boulder Falls while I was hanging out over there. He's very lucky he's alive. And this is some of our good times down in the filming. The film crew in the upper left is the Pioneer Productions, and they thought that safety was number about 50 on the list. And so when one of their people got sick, and Kenneth traded respirators and discovered that one of the rats where we were living had chewed a hole in her respirator, we told her we were done. And she was like, no, you're not. And we're like, yes, we're done. The other major part of my career has been looking at lava caves, which are, I think, one of the coolest caves in the world, because you can see the microbes. And I realize that these fungi don't really qualify as microscopic, but this is in the Azores. We developed a whole lot of really cool associations with colleagues in the Azores and in Hawaii. Lourdes Dubkovich and several of my grad students, and Fred Stone, who many of you may know, and Penny and Mike. So the microbes in these caves are truly beautiful. I think that they would make great artwork. So they come in a lot of different colors. They cover whole walls. They're pinks and oranges and yellows and whites. They can look like rumpled things. They can be lacy, and like in the bottom right for you, they are like wormies, little wormies. We were very excited when we found little wormies. And this is what they look like when you look at them in a scanning electron microscope. And what's really different here is instead of being sort of boring, sort of just round and rods and filaments, they're all adorned. Many of them have thread-like extensions, which are probably pili or fimbriae structures, but they look like just masses of medusas. All kinds of hairy structures all over them. We also discovered up in the upper left that there are a lot of beads on a string, which is one of the morphologies we see worldwide in caves. And in the middle, on the left, you'll see biofilm. We're back to snot. And those biofilm are covered with various rod-shaped bacteria in all kinds of folds. And then in the bottom left, we discovered our microchoia again. So we were able to extend their existence to also being in lava caves. This is Lava Beds National Monument, a photo my husband took, and you can see everything that's yellow there is a microorganism, and the whole wall is covered with these. And the bacterial diversity of these is truly amazing. And so we started studying different caves at lava beds. We were able to study caves that had human influence and caves that didn't have human influence. And we're able to ascertain that there's sort of almost a carrying capacity. Caves like Carlsbad Cavern show a lot of human impact. Caves like lava beds that have tens of thousands but not hundreds of thousands of visitors don't so obviously show the impact on the microbial communities. So that was one of the neat things about studying lava beds. We also discovered some morphologies that are really wildly different in lava beds, such as these hairy sausages that are budding off balloons on the end, almost like they're blowing up the balloons. And one of the things that I think is most significant is when I did the sequencing, I looked at samples from the surface, from above the cave, versus samples from in the cave. Because one of the things I wanted to know was the old dogma that cave microorganisms are just a subset of what's on the surface. Is this true? And what we discovered is that there's only a 14% overlap between the two sets of communities. So these are very different communities. And it turns out that one of the major groups in there are some nitrogen cyclers. So it's an environment that doesn't have a whole lot of nitrogen. So these guys are contributing to being able to feed the rest of the cave community. And we're learning a whole lot more about this and what's going on with this. We also have done a project called Microbes that Masquerade as Minerals, with the idea that if life exists on places like Mars or other extraterrestrial solar bodies in our solar system, it's likely to be microbial, and it may not be living. It may be buried in various mineral crusts. And so what we've done is look at some of these different varieties. In the upper left, you'll see one of my favorites. It's a copper silicate, chrysocolla. And down in the lower left was really cool. We discovered this in a tourist cave of all things. This is volcanic glass with little iron hexagons on it that weren't made microbially but are being eaten by microbes. We also found a lot of iron formations, such as you see on the right. And in particular, we've looked into a lot of what's going on with these copper ones, and it turns out that you can grow them, which Penny has done, and get them to precipitate copper. And if you look in the interior, they are full of microorganisms. And these are some of the actinos, but look at those long filamentous structures. There are microchoia, which is really cool. So the other thing is that we've turned our attention in the last few years to the impact that we humans have on the cave. And I've done some of these studies with Kathy Lavoie. I've actually swabbed cavers as they come past me in Lechuguilla Cave to see what microorganisms they carry. I've swabbed rocks in the cave to see. I've gone to poop spots and pee spots to see if I can recover E. coli. I've had one of them break and dump me into the pee spot in Lechuguilla. So fun field work. But you are essentially a walking microbial petri dish. You have 10 times the number of microbes in and on you as you have cells in your body. And so what these microorganisms do is we now know through research that we've done in the last 10 years, we know that they're part of what keeps you healthy. They're part of what gets you to develop as a baby. but they also are falling off you all the time. As you sit there, you're shedding 10,000 skin fragments a minute or more. Each one of those is a life raft for microorganisms that are on your skin. And so as you go through a cave, you're shedding all of that organic matter and all those microbes into the cave. So we did a lot of studies in Carlsbad Cavern. I think Kathy has done some in Mammoth Cave too, looking at how much impact do you see. We see a lot of impact in terms of fungi. You can pick up E. coli off the restroom floor in Carlsbad Cavern, but you can't pick it up off the trail, because I'm here to tell you the rangers do an amazing job of cleaning up after somebody poops on the trail. But you have never seen an angrier, grumpier person than a ranger on poop patrol. They are not happy. So this gave us an idea for the work on white nose syndrome. And this we have pursued with 2 bat biologists, a fungal biologist, a cave microbiologist, and cave microclimate people. Some of the people you'll recognize are Debbie Beecher, who has done all the holding of the bats. She's there in Via Luz in the upper area. Let's see if whoops, that was not the one to push. And she's up there in the middle in the pink shirt in Cueva de Via Luz. And so she basically captures the bats for me and is able to hold them so that we can swab them. And we basically swab all the membranes and the fur parts of the bat to get a baseline of who lives on the bat. So if you think about it, you have all those microbes on you that I've talked about, and they are able to do different things for you. But one of the things they do is they sometimes produce antimicrobials so that they can kill off things that come to try to harm you. And so we wondered, is it possible that the bats do the same thing? This is Debbie. As you see, one of the bats she's just releasing in Carlsbad Cavern. So we've been able to swab 13 different species of bats. And one of the major things we found, now I would have thought that these bats would differ in terms of the different bat species would have different microbiota, and that different regions would have really different microbiota. That's not so much true. What really matters is whether you were caught in the cave or you were caught on the surface. Because it appears as they hang there in the cave, they are acquiring various microorganisms. Just like as you're sitting in this room, you are acquiring new microorganisms from the person beside you, from the seat you're sitting on. Did you know that in restaurants, the dirtiest place is the seat you're sitting on? Did you know that when you kiss your significant other, you're acquiring 80 million microbes from a good intimate kiss? So you can think about whether you got a peck and only 10 million, or a good kiss and 80 million. So I don't think the bats are kissing, but there is good research to show that they are rubbing up against each other. They're putting dust on them of different colors, and they can tell who touches whom. And so here is one of the things we discovered, which is it depends on where you're caught. The other thing we're doing is we are culturing Actinobacteria off of these bats and testing them in the lab for whether they can kill Pseudogymnoascus destructans, the fungus that kills the bats in the disease white nose syndrome. And so what you're looking here in the center, the white, is an Actinobacteria, and it is growing to a nice stage, and then has had an overlay poured of food for the fungus, and then an overlay of P. destructans has been poured, and that cleared area is the area where our actino is killing the fungal pathogen. And we have 11 of these so far from 5 different bat species across Arizona and New Mexico that can inhibit. One of them is the rhodococcus that Chris Cornelison has been doing studies on, but the others are Streptomyces, and that's the genus that we get the antibiotic streptomycin from. So this holds promise of potentially an additional treatment that might be useful. So out of all these studies, what we have developed now is we have a much better idea of who's home. Now we want to know more about their function. What are they doing? So we're delving into the omics. So we're doing genomics and we're moving on to proteomics so that we can tell not just that the Actinobacteria are there, but what are they actually doing on the bat. And we're looking also at the microbial role in nutrient cycling. We and other researchers now know that it looks like bacteria in the cave are a major part of cycling nitrogen, possibly phosphorus. We know they also cycle sulfur. And so we're looking to see how do these microorganisms engineer the cave environment. So you can think as you go through your next cave about the fact that the walls are truly alive that you're looking at. We also still want to pursue, is it an indigenous subsurface microbiota, and do those microorganisms actually adapt to the cave environment? Are there troglodytic microorganisms? And we want to find out who the microchoia are too. So I want to end by saying a big thank you to my husband who's taken all the fabulous science in action shots and been able to enrich my talks over many, many years. And I have an amazing group of students in the lab. And a lot of this work just wouldn't be done without their efforts. They are often a joy. They can occasionally do some really things that scare the living daylights out of me. But they are mostly just lots of cool fun to work with. So anyway, and I think we have time for some questions. And I can actually see you. Back in the back. Those crickets are about, the Carlsbad crickets are much smaller than the ones at Mammoth. So for you, they're probably about an inch, their body's about an inch, and then the legs come out from there. Where is it? Shout it out.
Can we collect samples in the field in a simple way?
In terms of like bats?
No, the microbes.
Oh, the microbes. It depends on how careful you are, because one of the things we discovered is some of our Lechugia caves or samples are highly contaminated with Propionobacteria acnes, which comes off human skin, because it turns out the person who collected it didn't wear gloves. So the microbe samples, we have to preserve the DNA, which we have a solution to do, but you have to be really, really good at sterile technique, or otherwise I'm getting you in your cheese sandwich. Which I appreciate the idea, and I love getting pictures so we can see what's going on. Yes? Ah, okay. So in eating, you mean the organic matter, right? Not the rock stuff. So I've been concentrating more on the ones who eat the rock, so I know less about the ones who eat the organic carbon. I can tell you the crickets will eat just about anything, including diarrhea and vomit, and each other. But the microbes, a lot of the fungi are probably degrading it. We do less work with the fungi, but a lot of the fungi are major degraders, and it turns out we have a lot of fungi in our caves, including Cryptococcus, by the way, which is what causes cryptococcal meningitis. Penny.
Do you think that any of the fungi that we find are actually indigenous to caves?
So Penny wanted to know whether I think that the fungi that we find are indigenous to the cave, or are they all more surface ones. So this goes back to how we actually culture them, if we're trying to culture them. And one of the things that Larry, Mallory, and Penny have pioneered is the idea that when you go after organisms that are adapted to the cave, you have to use techniques that fit with that aren't for surface organisms. So most of the culturing we've done out of the cave has been more surface type stuff, but we started using a technique which is serial dilution, so that we can get rid of the weeds. And we're starting to find, for instance, the close relatives to Pseudogymnoascus destructans. We're able to culture those now. If you're patient, and like some of the cultures that Penny and I have done, grow for 2 years before you actually start getting the guys you're after. And if we don't feed them a lot, and in fact I have a student who has been working on 13-year-old Lechugia cultures. So if we use those same techniques in fungi, and in fact Larry, Mallory, and I did an experiment on that once and got very different things. I think they're there, but I think we haven't looked hard enough. Any other questions? Back in the back, I see two.
Where in our solar system do you think we'll first find microbial life indigenous to the
Well, so where in our solar system will we discover life? And I would have thought it would be Mars, but Europa's looking much better now. They're getting some really good things. And there's recent stuff on finding methane on Mars, which also is very promising. And we're ready to go. We're just a little too old. Back behind you. So at least some of it is precipitating chemically, some of it may be microbiologically. In Sulphur River in Parker Cave, you can actually see granules of elemental sulfur in the microbial bodies. And it's sort of like we store fat, they're storing it because there's still energy to be had from further oxidation of it. And so some could be chemicals, probably a lot of it is chemical, some could be microbiological. Any others? Thank you very much for coming today.
Thank you very much, Diana, for a fascinating talk. I think it's remarkable indeed that some of the most famous cave scientists in the world are amongst the members of our society. So when you see Kenneth and Diana this week, please thank them for their excellent work and for participating in our conference. I hope to see you tomorrow at 1 o'clock in this very room where C. William Steele will give a luminary presentation. Thank you for coming. Enjoy the rest of your day.
Thank you. That was fun.
Good job. Thank you. Fabulous.